Friends, welcome back to my channel, and today we are going to study about lipids part 3, and in this video we'll study about structural lipids, glycerophospholipids and ether-linked phospholipids. So first, structural lipids. Structural lipids means they form the membrane bilayer. Now, as you all know, the membrane is a phospholipid bilayer. Now, this phospholipid bilayer contains polar head and non-polar tails. These non-polar tails faces each other, whereas the polar head faces the surrounding aqueous environment. This gives the phospholipid membrane a distinct structure. Now, there are 5 general types of lipids that constitute the membrane. These are glycerophospholipids, that means a phosphate group will be there. Lipids, of course, it is there, and a glycerol backbone. Next is galactolipids and sulfolipids. That means they contain galactose unit. In sulfolipids, they have a sulfur unit. The backbone is again glycerol. Archibacterial tetraether lipids. They are also based on glycerol backbone, but they contain ether linkage. Sphingolipids, that means here the backbone is sphingosine. And next is sterols. So these are the 5 general types of membrane lipids. Apart from that, we have phospholipids and glycolipids. Actually, glycerophospholipid is a type of phospholipid only. Similarly, for glycolipids also, this galactolipid is a type of glycolipids only. But these are mentioned separately because apart from glycerophospholipids, the phospholipids contains much more variations. So this flowchart tells you in general about all the membrane lipids. So we have 2 types of lipids. We already studied about storage lipids, and now we are studying about membrane lipids. So storage lipids, we already studied. It is triacylglycerol, that is the backbone is of glycerol, and all the 3 carbons are attached with fatty acids. Coming to membrane lipids, we have 3 categories, that is phospholipids, we have glycolipids, we have archibacterial ether lipids. Phospholipids means it should have a phosphate group. Glycolipids means it should have carbohydrate. Archibacterial ether lipids means they are ether linked. Rest all other lipids are ester linked. Now phospholipids are again of 2 types, that is glycerophospholipid, that means the backbone is of glycerol, or sphingolipid, that means the backbone is of sphingosine. Similarly, glycolipids can also be of 2 types. One is sphingolipid, again the backbone of sphingosine, and galactolipid or sulfolipids where the backbone is of glycerol. So basically for any membrane lipids, the backbone should be either of glycerol or sphingosine. For all the phospholipids where the name sphingolipid will come, there you can understand by the name sphingo, that means the backbone is of sphingosine. Rest all other membrane lipids contain the glycerol backbone. In case of glycerophospholipids, by the name only you can able to tell that it is having a glycerol backbone. But in case of galactolipids, there the glycero word is not there. Okay, so always remember that the backbone can be either of glycerol or it can be sphingosine, but mostly it is glycerol only. If it is sphingosine, then the name will tell you that the backbone is of sphingosine by the name sphingolipid. Otherwise, for rest all cases, the backbone is of glycerol. Now first we will study about the glycerophospholipids. Now this is the structure, general structure of glycerophospholipid. That means the backbone is glycerol. Now we already studied the structure of glycerol. This is the structure of glycerol. So with the first carbon, fatty acid is attached. With the second carbon, fatty acid is attached. With the third carbon, the name is phospholipid. So this phosphate group is attached. And with this phosphate group, any alcohol will be attached. So the name phospho means from phosphate. So with the third carbon, we have a phosphate attached and then we have an alcohol attached. That means in first and second position, we have ester linkage, and in the third position, we have phosphodiester linkage. Same it is written here. Now, here, in case of glycerophospholipid, the parent compound is phosphatidic acid. This how it looks. This is the glycerol backbone, carbon number 1, carbon number 2 and carbon number 3. With carbon number 1, fatty acid, esterified. Carbon number 2, again fatty acid, esterified. Carbon number 3, we have a phosphate and an alcohol. So this alcohol is written by X group. This forms the head group. That means this is polar because alcohol is polar, and the phosphate also, phosphatidic acid also. Correct. So this is common. Now this X group will be different, and based on the X group, the name of glycerophospholipids will also differ. Now one more thing you can note, generally in glycerophospholipids, the first fatty acid attached to C1 of glycerol is a saturated fatty acid, and the second fatty acid attached to C2 of glycerol is an unsaturated fatty acid. So mostly you will see this structural makeup. That means with C1 of glycerol, we'll have saturated fatty acid, and with C2 of glycerol, we will have unsaturated fatty acid. And the third position, we have phosphate group attached with polar head group, that is any alcohol. So these linkages are ester linkages, and because here phosphate is present, so this linkage is a phosphodiester linkage. Now, if that X, that means the polar head group, rest all structure will remain the same. If the X is H, then we call it as phosphatidic acid, whose net charge is -1 at neutral pH. Similarly, if the X group is ethanolamine, then the name will be phosphatidyl ethanolamine. The ethanolamine is positively charged, making the net charge 0. Why? Because phosphate group is present, which contains negative charge, and this positive and negative will neutralize each other. If the X is choline, then the glycerophospholipid name will be phosphatidyl choline. Similarly, the net charge is given, and from here you can see the charge present on this X group, and one charge is present negative already because phosphate is present. So that negative and this positive is there, it will neutralize each other. If both negative are there, the net charge will be negative. So like that it is. Clear? If the X group is serine, the glycerophospholipid will be known as phosphatidyl serine. Similarly, if glycerol is present, it will be known as phosphatidyl glycerol. Similarly, if myoinositol biphosphate is present, the name will be phosphatidyl inositol biphosphate. If phosphatidyl glycerol is present, the name is cardiolipin. So these are the varieties of glycerophospholipids which differ in the X group. So the name of the glycerophospholipid will depend upon the type of X group present. Now some phospholipids have ether-linked fatty acids. Now some animal tissue and some eucellular organisms are rich in ether lipids. For example, some vertebrates, halophilic bacteria, ciliated protists, and certain invertebrates. So the example of ether-linked fatty acid in vertebrate, one is plasmalogen. This phospholipid is present in our heart. So about half of the heart phospholipid are plasmalogens. So the ether-linked fatty acid is present in the phospholipid found in our heart. The name is plasmalogen. Similarly, one more example is platelet activating factors. It also contains ether-linked fatty acids. So platelet activating factor, from the name you can understand that it activates platelet. So that means these are potent molecular signal. They release from basophils. They help to aggregate platelets. They help to release serotonin from platelets, and they have an important role in inflammation and allergic responses. So that's all for glycerophospholipids. Now, one question from my end for you all, that is a common procedure for cleaning the grease trapped in a sink is to add product that contains sodium hydroxide. Explain why this works. Sodium hydroxide means NaOH. So this is present. So think accordingly and let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.